Good morning, my dear student. On the behalf of KTM College Nasik and Department of Chemistry of this college, I am Dr. S.R. Labhade welcomes you to the lecture on polarographic analysis. Student, in previous lecture we discussed the instrumentation of polarography, that is a apparatus used in polarographic analysis. Today we discuss the working of polarography. Student, we know that polarography is a linear potential scan voltammetric method of qualitative and quantitative analysis performed in a quiescent analyte solution using polarized dropping mercury working electrode and pool of mercury counter electrode. Therefore, with suitable example, we can discuss here the working of polarography. Working of polarography. In polarographic analysis, a sample solution is taken into polarographic cell containing dropping mercury electrode and pool of mercury counter electrode. Therefore, sample solution, particularly analyte solution, is taken into the polarographic cell. Actually, it is the electrolysis cell. When we use DME and pool of mercury as a electrode, then it becomes a polarographic cell. In this case, DME is made cathode, means negative terminal of battery is attached, and pool of mercury is made anode. That is a positive charge connected to the anode. Now here we can represent this polarographic cell. Initially here we have this container, and in this container there is a mercury, that is a pool of Hg. This is a positively charged electrode. A platinum wire is inserted and a positive potential is attached to this electrode. Second electrode is dropping mercury electrode. We can simplify these in this fashion. There is again a reference electrode if it is required to control the potential of this dropping mercury electrode, and this is a saturated cadmium electrode. This DME is attached to negative terminal and therefore it is act as a cathode. This is anode. Here there is inlet for bubbling a nitrogen gas, and this is outlet for that nitrogen gas, and this is a apparatus, that is a polarographic cell used in electrolysis of analyte by using dropping mercury electrode and pool of mercury. Now in this cell a analyte solution is taken, and sometimes a supporting electrolyte supporting electrolyte is also added in this analyte solution, a sample solution or analyte solution. This is a site for that redox reaction. This is the representation of polarographic cell. This is a working electrode DME and pool of mercury as a counter electrode. Mercury is present at the base of the cell, and here small spherical drop of mercury are generated on the tip of the capillary. That is a site for redox reaction. Now with example of cadmium ion, we can discuss here how the polarography work. Suppose for example, cadmium 2 plus ion, the cadmium 2 plus ions are taken into polarographic cell. That means here we have to analyze cadmium ion, therefore polarographic cell. We know that if oxygen is present in a polarographic cell, then it is reducible at dropping mercury electrode, and that reduction current is get added into the reduction current of analyte, and therefore in polarographic analysis it is essential to remove the dissolved oxygen from the analyte solution, and it is done by passing the nitrogen gas or by bubbling the nitrogen gas into the analyte solution. Therefore nitrogen gas expels out the dissolved oxygen from the sample, and nitrogen gas is very much inert, if present in the solution does not affect the measurement. Therefore after that the dissolved oxygen is removed from analyte solution by bubbling the nitrogen gas. Therefore nitrogen gas is removed by bubbling the dissolved oxygen can be removed by bubbling the nitrogen gas through this inlet, and here it will be mixed with analyte solution, and then oxygen or extra nitrogen gas is given out of this outlet. In electrochemistry, the analyte is transported to the electrode surface by three different phenomena, and that is, first is called as convection, second one is electromigration, and third one is diffusion. Therefore first, that is a convection. Convection is a mechanical transportation of ion to the electrode surface, and it is done by stirring the solution. Solution is stirred or it is mixed. At that time these ions are mechanically transported to the electrode surface, and that phenomenon is called as convection. Second thing is that, or second phenomenon is called as electromigration. Electromigration, that is a migration under the influence of electric field. Means here positively charged ions are attracted towards the negatively charged electrode, and negatively charged ions are attracted to the positively charged electrode because of the electrostatic force of attraction, and that migration is termed as a electromigration. Third type of migration or third type of phenomenon that transport the ion to the electrode surface, that is called as diffusion. Diffusion, that is a transportation of ion to the electrode surface by creation of concentration gradient, and this transportation is called as diffusion. Now, what is this concentration gradient and how these ions are transported to the electrode surface? Now here, suppose for example we have this electrode. Suppose this is a mercury electrode. This is hanging into the analyte solution. If it is a negatively charged, and here initially if this electrode is get polarized, these positively charged ions are get migrated to this electrode surface and those get deposited onto the electrode surface. Therefore, in this region, the number of ions, analyte ions, are found to be less than the ions present in the bulk of the solution. Therefore, we have two regions here. In this case, here we have a region with less or no ion, and here we have a region that containing excess of ion. Therefore, here in the same solution, we have two different layers of the solution having different concentration, and therefore here this is called as creation of concentration gradient. When ions are migrated from the region of higher concentration to region of lower concentration, that process is called as diffusion. That is a migration of ions from the region of higher concentration to the region of lower concentration, and this is called as concentration, and this is called as diffusion. And therefore here in polarographic analysis, diffusion is the phenomenon that controls the transportation of ion to the electrode surface. Therefore here in electrochemistry, ions are transported to the electrode surface by convection phenomenon, by electromigration phenomenon, and by diffusion phenomenon. But in polarographic analysis, convection is not possible because we are performing polarographic analysis in a quiescent analyte solution, that is an unstirred analyte solution. Therefore, we are not studying the solution. That is why a convection phenomenon is not possible in polarographic analysis. Electromigration and diffusion are the two phenomena made possible in polarographic analysis for transportation of ion to the DME electrode. Now here we discuss how actually this transportation takes place. Suppose for example when we linearly increase the potential of the working electrode and this counter electrode, this is a counter electrode and this is a working electrode, then what happens that we study here. When we apply linearly increasing DC potential across DME and pool of Hg, then Here we are applying the potential across these two electrodes, DME and pool of mercury. Here surface area of pool of mercury is very high compared to the surface area of this mercury drop generated on the tip of dropping mercury electrode. Therefore here with application of this potential, the dropping mercury electrode is get polarized due to very high current density. Current density is the magnitude of current divided by surface area of the electrode. Because of very high current density, the dropping mercury electrode is get polarized and it acquires negative charge. As electrode is become polarized, it acquires negative charge and then once the electrode acquires negative charge, the positively charged cadmium 2 plus ions are get migrated to mercury drop. Therefore here, as this electrode is negative charge, cadmium ion which is near to electrode surface that are first get migrated, and when potential reaches to sufficient high, then there may be a reduction of this cadmium ion, and here the cadmium ion which is present in this region of solution and cadmium ion present in the bulk of this solution, that creates a concentration gradient. Again here, this cadmium ion migrated to this region because of the diffusion process. Therefore here, the positively charged cadmium ions are get migrated to the mercury drop by electromigration and diffusion phenomenon or diffusion process. And here the total current flowing through the cell is equal to I is equal to Im migration current plus Id diffusion current. Therefore here current flowing through the cell, that is the sum of migration current and the diffusion current. Now here in polarographic analysis is carried out in a quiescent analyte solution. Therefore there is no convection current, that already we discussed. Therefore in this case, when potential reaches equal to the reduction potential of analyte, this potential is called as decomposition potential. Applied potential when it is reaches equal to the reduction potential of analyte, that is called as decomposition potential. Then the deposition of analyte takes place. As analyte deposited onto the electrode surface. Suppose for example cadmium in place of mercury plus 2 electron, it gives cadmium amalgam CdHg. Therefore here this reaction taking place on the surface of mercury drop, and that mercury is given at the base of this cell. Again new drop is generated, and here we have the reaction on that new drop. This process is continuous, that means the analyte is present in the solution. Now here as analyte is deposited onto the electrode, the resistance of solution increases, and to overcome this difficulty, and excess of indifferent electrolyte, that is called as supporting electrolyte, is added in polarographic cell. Therefore here analyte solution plus supporting electrolyte, that is taken into the polarographic cell. Indifferent or supporting electrolyte is that electrolyte whose concentration is 10 times more than the concentration of analyte, that is added in a polarographic cell to nullify the resistance of this cell as well as the migration current. Therefore here the indifferent electrolyte means that is the electrolyte which is not reducible at a potential where analyte reduces. This indifferent electrolyte decreases the resistance of cell and it eliminates the migration current. This eliminates the migration current, and at that time the current flowing through the cell I equal to only ID. Because addition of excess of supporting electrolyte decreases the migration current as well as it decreases the resistance of the cell. Therefore, total current flowing through the cell is equal to only ID. ID means diffusion current. In a polarographic analysis, total reduction process is controlled by the diffusion process. In polarographic analysis, the reduction process is controlled by the diffusion process of the analyte. And so the current measured in this case is called as diffusion current. Therefore, in polarographic analysis, diffusion current is measured for quantitative analysis. Here, there is no convection current, there is no migration current. Analytes are transported to the electrode surface by diffusion process, and therefore that current is measured as a diffusion current. This diffusion current is because of the following reduction reaction taking place on the surface of the mercury drop. Here, in this case, cadmium 2 plus ion in presence of Hg plus 2 electron, it gives cadmium amalgam. As analyte reduces onto the electrode surface, electrons are required. These electrons are provided from the dropping mercury electrode, and therefore there is flow of electron. As flow of electron is observed, we observe the current. Therefore, when this reaction taking place on the surface of electrode, we observe increase in current. Then, as the reduction of analyte takes place, the increase in current is observed. The increase in current is observed, and that will be reaches to maximum value, and that reaches to maximum value, and then becomes constant, means analyte is completely reduced. Analyte is completely reduced. Therefore, here due to this reaction, there is flow of current. Flow of current is observed till this reaction is taking place on the surface of mercury drop. When analyte is completely reduced, there is no cadmium ion in the solution. There is no such type of reaction. At that time, current becomes constant, and we say that the reaction is completed, means analyte is completely deposited. If we are still increasing the potential of working electrode, that is dropping mercury electrode, then we observe that there is another reaction. If the potential of DME is increasing, then the reaction of supporting electrolyte takes place, and then we stop the analysis. Therefore, signal incompletion of reaction is because of the reaction of supporting electrolyte. As that analyte is reduced completely, reduction of supporting electrolyte takes place, and then we can say that we have to stop the electrolysis process. In polarographic analysis, the reduction current due to analyte, that is called as diffusion current. Reduction current due to the analyte is measured for quantitative analysis. And this current is directly proportional to concentration of the analyte which undergo reduction at dropping mercury electrode. Then the concentration of the analyte in the sample solution is determined by using a Ilkovic equation, and this a diffusion current. The concentration of the analyte is determined by using diffusion current and Ilkovic equation. This Ilkovic equation is useful for the determination of analyte in the sample solution. The equation is like this. Therefore, ID is equal to 607 into N into D raised to half into M raised to 2 by 3 into T raised to 1 by 6 into C, where this ID is potential dependent diffusion current, and it is measured in microampere. 607 is constant, N is number of electrons involved in redox reaction. For cadmium it is 2 redox reaction. Then here D is diffusion coefficient of the analyte. Diffusion coefficient, and it is measured in square centimeter per second. M is mercury fluoride, and it is measured in milligram per second. T is drop time, and we know that it is measured in second. C is concentration of analyte in millimole, and 1 millimole is equal to 10 raised to minus 3 mole. Therefore, in polarographic analysis, concentration can be measured in millimoles level, that is 10 raised to minus 3 to 10 raised to minus 4 moles per liter. Now, in this way, the concentration of the analyte is determined by using this Ilkovic equation, and we have to solve different problems based on this Ilkovic equation for determination of concentration of analyte, or for determination of number of electrons involved in redox reaction, or diffusion coefficient, or mercury fluoride. Student, you can find out the answer of the following question through this discussion. Number 1 is define decomposition potential. Second one is define supporting electrolyte. And third question, explain with suitable example the working of polarography. Explain the working of polarography. Student, you can find the answer of this question. With this, I invite you to my next lecture on polarographic analysis.